What if something could control your mind without controlling your body? Why would a cockroach follow a wasp into a dark burrow when it can still escape? What could make an animal lose the desire to run while its legs still work? Can a tiny wasp really switch off another animal's will to escape? And what exactly happens inside its brain? The cockroach can still run. Her legs work. Her brain works. What has been removed is the desire to leave. She follows the wasp into the burrow. She sits down. She waits. The wasp is a quarter of her size. It has no leash. It is not threatening her. It is holding one of her antennae, lightly, the way a person might hold a child's hand walking into a dentist's office. The cockroach has every physical capacity to escape. She does not. This is the most precise act of mind control in the natural world. And the science of how it works, when you read it carefully, is much stranger than the horror story it usually gets reduced to. What did the wasp do to her? Why doesn't she run? Here is what you need to know about the jewel wasp cockroach mind control sequence. The wasp is Ampulex compressa. She is roughly 2 centimeters long, an iridescent metallic green, and she is one of about 200 species of parasitoid wasps that use other insects as living hosts. Her target is the American cockroach, Paraplaneta americana, an animal 3 or 4 times her size and considerably faster. A direct fight would go badly for the wasp, so she does not fight. She performs surgery. Over the course of about a minute, she will deliver 2 precisely targeted stings into the cockroach's central nervous system. Not random stings, targeted ones. Each one is aimed at a specific cluster of nerves with a specific job, and each one removes a specific capacity. By the time she is finished, the cockroach is not paralyzed. She is not unconscious. She is not in pain that we can measure. She has simply lost the will to leave. The wasp then takes hold of one of the cockroach's antennae and leads her, walking on her own 6 legs, into a burrow the wasp has prepared. The wasp lays a single egg on the cockroach's abdomen. She seals the burrow. The cockroach sits there. For days. What does it mean exactly to remove only the will to walk? To leave a body intact but cancel the thing inside it that wanted out? The answer, when you trace it through the research, is much more specific than mind control sounds like. Where, precisely, in the cockroach's brain is the will to walk located? What does the venom actually do? Here is what we now know about the mechanism, thanks largely to 2 decades of work by the Israeli neuroethologist Frederick Libersat at Ben-Gurion University of the Negev. The 2 stings are not equivalent. They do different things in different places for different reasons. The first sting goes into the cockroach's thoracic ganglion, a cluster of nerves in the chest that controls the front legs. The wasp injects venom directly into this site. Within seconds, the cockroach's front legs go limp. The paralysis is temporary, lasting only a few minutes. It is not what subdues the cockroach for the long term. Its job is much narrower. The first sting exists so that the cockroach cannot fight back with her front legs while the wasp lines up the second sting. The second sting is the important one. The wasp climbs onto the cockroach's head and pushes her stinger between the cuticle plates, threading it directly into the head ganglia, specifically the subesophageal ganglion and the supraesophageal ganglion, the 2 clusters of nerves that, between them, manage the cockroach's central complex. The central complex, in an insect brain, is what neuroscientists call a locomotory command center. It is the part of the brain that initiates and maintains walking. The wasp stinger, it turns out, can feel its way to the right spot. In 2003, Libersat's team showed that wasps stinging a brainless cockroach took 15 times longer to deliver their venom, meaning the stinger uses sensory feedback from the prey's nervous tissue to confirm it has reached the target before releasing the venom. This is parasitoid wasp behavior manipulation at a precision most neurosurgeons would envy. What happens next is what everyone remembers. For roughly 30 minutes, the cockroach grooms herself. Compulsively, obsessively, she cleans her antennae, her legs, her wings. The team in Beersheba showed in 2010, in a paper in PLOS 1 titled "A Wasp Manipulates Neuronal Activity in the Subesophageal Ganglion to Decrease the Drive for Walking in Its Cockroach Prey," that this grooming is driven by dopamine in the venom. The wasp is, in effect, switching on a grooming circuit by chemical injection. Then the grooming stops, and the cockroach enters a state called hypokinesia, which lasts between 3 and 7 days. Hypokinesia is not paralysis. The cockroach can stand. She can walk if she has to, but her drive to self-initiate walking, to decide on her own to leave, is reduced to almost nothing. This is the part Libersat and his colleague Ramgal mapped most carefully. The subesophageal ganglion is not sending its normal regulatory signals to the central complex. The cockroach's locomotion center is intact. It has simply stopped getting the green light from the part of her brain that would normally say go. She can leave. She just no longer wants to. What is the difference between being unable to leave and being unable to want to leave? Is there one? Many people think the Jewel Wasp story is a horror story about zombies. A small, beautiful animal turned into a puppet by a smaller, even more beautiful animal, all the way to her own grave. The research, when you read it carefully, is something less theatrical and more uncomfortable than that. The cockroach is not a zombie. She is not a puppet. There is no string. The wasp does not control her in real time. After the burrow is sealed, the wasp is somewhere else doing wasp things. The cockroach is alone. She has full motor function. Her sensory systems are intact. She can detect light, vibration, chemicals in the air. She knows, in whatever sense a cockroach knows things, that she is in a small dark space. She does not leave. What the wasp has done is not control. It is something more precise. The wasp has performed a chemical removal of one specific psychological function, the drive to initiate escape, while leaving everything else operational. The cockroach is not missing her body. She is missing the part of her decision-making system that, in an unstung cockroach, would say get out of here. This is the part that makes biologists nervous. Because the drive to escape, the drive to leave, the drive to move on, this is one of the deepest and most ancient circuits in any nervous system. It is not a learned behavior. It is not a skill. It is part of what makes an animal an animal. In 2018, Arvidsson, Landa, Frankenberg, and Adams published a detailed life history paper on the jewel wasp in the Journal of Hymenoptera Research, walking through the entire sequence from sting to burrow to larval feeding. The whole reproductive strategy depends on this one fact. The cockroach, with every capacity she had before, no longer wants to use them. There is no scientific consensus on whether the cockroach is suffering. Libersat himself has written carefully that we cannot know what the experience is like from inside. What we can say is that the behavioral signature looks unsettlingly familiar. An animal with everything it needs to escape, sitting in a small dark space, not escaping. And in 1972, the American psychologist Martin Seligman gave that pattern a name. He called it learned helplessness. He had not, of course, been studying cockroaches. That is the part the horror coverage usually misses. The wasp did not invent a behavior. She found a circuit that already existed in the cockroach, and she found a chemical key for it, and she turned it on. The neuroethology of escape behavior is a story about how thin the wall is between the will to leave and the chemistry that holds it up. How thin is that wall in us? What turns the same circuit on without a sting? Here is the central question, then. The wasp can turn off the cockroach's drive to escape with a precise drop of venom in a specific location, lasting between 3 and 7 days. Humans do not get stung by jewel wasps, but humans sometimes end up in the same place, sitting in a situation we could leave, capable of standing up, not standing up. The wall between agency and immobility is, it turns out, more chemical than we usually admit. The same brain circuits that the wasp targets in the cockroach locomotor command center's neuromodulatory inputs, the dopamine signaling that gates motivated movement, exist with different names and shapes in the mammalian brain. In humans, when motivation collapses, what collapses is not usually the body. It is the part of the brain that issues the signal to go. The point here is not that humans are cockroaches. The point is that the drive to leave is not a permanent feature of being alive. It is a function. It is performed by specific neural machinery, can be turned down, can be turned off chemically by a wasp. It can also be turned off by other means, by chronic stress, by social conditions, by repeated experience of trying to leave a situation and discovering that leaving does not help. That last condition is what Seligman documented in his 1967 and 1972 papers on learned helplessness. Animals subjected to escape attempts that consistently fail eventually stop attempting to escape, even when escape becomes possible again. The circuitry for action remains. The circuitry for initiating action shuts down. This is the uncomfortable parallel. The wasp uses venom. The world sometimes uses repetition. The mechanism at the level of neural systems is not as different as the two methods suggest. Both of them target the same approximate place in the brain. Both of them produce the same approximate outcome. The jewel wasp cockroach mind control story is, in this reading, not a story about insects. It is a story about how compliance gets manufactured in any nervous system that has the relevant circuits. The cockroach in the burrow, sitting still, is doing something we have a name for in human psychology. She just got there by chemistry instead of by experience. The legs work. The brain works. The wall has thinned out from inside. How do we tell the difference in ourselves between a situation we cannot leave and one we have stopped trying to? There is something to sit with here. The cockroach in the burrow is not a metaphor. She is a real animal, alive, hypokinetic, capable of walking and not walking. The wasp's larva will eat her, slowly, from the inside, over the course of about a week. She does not know this. By every measure of insect neurobiology we have, she is not afraid. The chemistry that would normally make her afraid is not signaling. She sits. What is uncomfortable about this story is not the cruelty of the wasp. The wasp is a wasp. She is doing what wasps do with extraordinary precision, for the benefit of a single egg. What is uncomfortable is the discovery, sitting at the bottom of the neuroethology, that the cockroach's will to leave was always a thing that could be turned off. It was held in place by chemistry, and the chemistry can be reached. The same is true in different ways in different brains with different keys for every nervous system that has the relevant circuit. The cockroach did not lose her body. She lost the part of herself that wanted to use it. So can we? How does an animal or a person get the wanting back? Where is it stored? The jewel wasp turns off the cockroach's drive to leave by injecting a chemical. There is another insect, a honeybee in a laboratory in Newcastle, whose drive to engage with the world can be turned in the opposite direction, also chemically, also measurably, by something much closer to ordinary stress. What that experiment looks like and what it suggests about how easily any small brain can be tipped from optimism into pessimism is the next thing we will look at.